this section, we're talking about water microbiology. Water has to be tested before it can be okayed or before it's declared use by humans, that it's safe for human consumption. And sometimes they will test water and they will say it's not safe for human consumption, but you could use it, say, to water a golf course or whatever. So if it's potable water, it's safe for human consumption. Non-potable water, don't drink it. This particular exercise deals with a type of test that's called the presumptive test. We often refer to the most probable number. It's an estimate of how many bacteria are in the water sample. So when you're testing water, what we often are looking for are coliform bacteria. These tend to be gram-negative bacteria that are naturally found in the intestines. And so if you see those in the water sample, it's an indication that there's been some fecal waste contamination. In other words, don't drink the water. So most of your testing procedures are geared towards testing for those coliforms. Some places, there's a couple ways you can run the test. Typically in the U.S., the main indicator organism they're looking for is E. coli, because that would indicate recent fecal contamination. So the traditional water testing method, there's 3 phases of it. There's a presumptive test, confirmed test, and a completed test. You would do it in that order to determine if the water is safe for humans to drink. So the presumptive test, this is the first stage of it, the first phase. And what you are looking for are lactose fermenters that will produce both acid and gas, because that's what the coliform bacteria are capable of doing. The most probable test, or MPN, determines, as I said at the very beginning, an estimate of the amount of contamination. You're not going to actually be counting the number of colonies like on a petri dish. You're going to be inoculating test tubes and coming up with a number, as you'll see in a moment. And so that's why it's an estimate. If you have negative results, then you do not have lactose fermenters. You would say the water is safe to consume. If you have positive results, then you have to move from the presumptive test to the next stage. The next stage would be your confirmed test. In this case, the water that you got the positive results on from the presumptive test, you would use that water sample now to inoculate a MAC plate, a McConkey plate, and EMB, which is eosin methylene blue plate, to determine if the bacteria present are gram-negatives. If you remember, a McConkey plate and the EMB plate, these are selective in that only gram-negative bacteria will grow on them. They're also differential media, but they are selective. So if you had a positive for the presumptive test, you inoculate your MAC and your EMB plates. If there's growth on there, then that confirms that it's a gram-negative bacteria. So, as I said earlier, your main concern is looking for the coliform bacteria, which are lactose fermenters. They are gram-negative. Now, sometimes you might have some gram-positives that are lactose fermentation positive, so they might have given a positive result in the presumptive test, but they would not grow in this confirmed test. If you had growth on the McConkey plate and the EMB plate, so now you've got, okay, from the presumptive test, you've got positive lactose fermenter. From the confirmed test, you've got gram-negative. Now do the completed test, which is simply take the samples and do a gram stain to confirm that it is indeed gram-negative. So with the initial presumptive test, which this particular experiment is dealing with, we take a water sample. We have 3 different sets of lactose tubes where you test for the lactose fermentation. And this first set over here with the higher amount of volume in it, this is what we call, well, all of them are lactose. We have different strengths of the lactose. This is a double strength, and in this one we add 10 mils of a sample, the same water sample. In this set of tubes we've added 1 mil, and in this set of tubes we've added 0.1 mil. So you have to be careful with your labeling to keep your tubes straight. You let them incubate. The lactose fermentation tubes have phenol red as a pH indicator in them. So when it is uninoculated and at a neutral pH, they are red. When acid is produced because of the pH indicator in there, it will turn it yellow. So as you can see, all of these tubes here are yellow. All of these are yellow. 2 of these here are yellow. 3 of them have not changed to a yellow color yet. They also have an inverted tube. If you look closely, you can kind of see this inverted tube in here. We call that a Durham tube. Before inoculation, that tube is filled with liquid. And so if gas production occurs, the gas being produced is going to be trapped, and it forms an air bubble in those inverted tubes. So it's very easy to see. If there's a gas bubble there, then it's positive for the gas. So after incubation, you simply go through and you look for acid and gas production. If it's positive, you record that number. So in this set of the 5 tubes, all 5 were positive. So you record the number 5. In the second set, all 5 of these tubes are positive for acid and gas. So you record, once again, 5. Over here, only 2 of the tubes were positive. So you record the number 2. Now what you do with this number, 552, there are charts, which by the way, nobody's memorized the charts. That's why you have them. You go to the chart and you look up the number 552, and it will give you another number that is the estimate of how many bacteria would be present. Usually it's recorded in per 100 mils of water. So like I said, it's just an estimate. This is the first test, just to run to see, is the water safe for human consumption or not? Because you got positive results here, you would have to then go to the next stage to confirm this.